Hello, this is Mrs. Kohler and welcome to another wonderful podcast. Today we are going to talk about enzymes. Here's your essential question. Make sure you pause this and write this down. The question for today, the essential question that has to do with this is, how are enzymes like a matchmaking service? So, first of all, what is an enzyme? Well, an enzyme is a special type of protein. So we already learned what proteins are. So the monomer of a protein is an amino acid, and the polymer of a protein is a polypeptide. So the good thing is you don't have to remember anything new really for enzymes. You just have to remember that it's a protein, so it's got the same monomers and polymers. So what exactly do enzymes do for us? Well, the first thing you need to understand is what a catalyst is. A catalyst is a compound that speeds up chemical reactions, and we need these in our body to make reactions happen quickly enough to keep us alive. And they do this by lowering activation energy. An enzyme is a specialized type of protein that acts as a catalyst in our bodies. So the enzyme is a type of protein that's going to speed up reactions within our bodies. And that allows the reactions to occur at a normal temperature, because for most reactions to happen, you have to heat them up. And you can't heat yourself up too far or you'll die. So the enzymes allow things to happen at our normal body temperature. So activation energy then is just the energy that you need to start a chemical reaction occurring. So every chemical reaction needs some kind of energy put into it, and usually that's heat. And like I said, you can't really heat our bodies up or we'll die. It would damage our proteins. So here's a diagram to kind of show you what's going on. So the reactants, this line with reactants, is where the level of energy that the things going into the reaction start at. Normally, it requires quite a bit of energy to get the reaction started. But if you use a catalyst or an enzyme, you're able to start it with a lot less energy put in, so that you're able to do this at a much lower temperature. And that's what enzymes do for our bodies. They let the reactions occur at a lower temperature. So that's what enzymes do for our bodies. So how exactly do they work? The mechanism they use is usually referred to as a lock and key mechanism. And it's called lock and key mechanism because just like there's only one key that will open any particular lock, one shape of key, there's also only one shape of molecule that will fit into each enzyme. So for an enzyme, there's one or sometimes more molecules that actually fit into it into a specific spot that's the correct shape, just like a key fits into a lock. The substrate is the reactant that the enzyme works on. In other words, that's the key part. That's the thing that's going into the enzyme. And the active site is the spot on the enzyme where the substrate fits into it. So that's the lock part. So let me show you a couple diagrams so you can understand what's going on. This bottom one here shows a key with a particular shape fitting into the lock. And next to it, it's showing a molecule with a certain shape. And you can see that the active site of the enzyme has the same shape as the molecule. So up here, you're just seeing a more realistic drawing of that. So this is an actual protein found in the human body and the substrate that goes into it. And you can see that it fits in there really nicely. That's the key fitting into the lock. And then this diagram is showing the whole process. It's showing the purple part is a substrate, the green part is the enzyme, so they join together where they're the same shape, and then they actually break apart into two. And the enzyme is the thing that actually helps it break apart without too much energy. Some examples of enzymes in our body. Usually when you hear a word that ends in ASE, that's usually going to be an enzyme. So lactase kind of sounds like lactose, which is a sugar that's found in milk. We use the enzyme lactase to break down lactose in milk, and people that are lactose intolerant don't have the enzyme that they need in order to break down the lactose. So if someone's lactose intolerant and they want to eat, say, ice cream, they'll take a little pill, and that pill has lactase in it. It's an enzyme that helps you digest the milk. DNA helicase is an enzyme that you're going to hear about later this year. It helps during DNA replication. It unzips the DNA. Sucrase kind of sounds like sucrose, and sucrose is table sugar. It's like the sugar you would put in your coffee. And this takes the sucrose and breaks it down into smaller parts so that our bodies can use it. So anything that ends in ASE is usually an enzyme. Now there are limits to what enzymes can do. They can only function at ideal temperatures. So there's a certain temperature range where the enzyme can function, and if it's too warm or too cold, it's not going to be able to function properly. And the 3D structure is really specific. So there's only certain molecules that can actually fit into an enzyme. So you need all kinds of different enzymes in your body to do all kinds of different jobs. There are certain molecules that can actually pretend to be the substrate, and they'll actually block the active site of enzymes. And this sounds like a bad thing, but we use these sometimes in medicines. They do prevent the enzymes from acting properly, but there's things called SSRIs. They're selective serotonin reuptake inhibitors. These are antidepressants. So when your brain wants to send a chemical signal, people with depression, the chemical signal doesn't make it to where it's supposed to go. It bounces back to the original cell. And then the messages aren't being sent properly, and that's what causes depression. When someone takes an SSRI, the SSRIs are almost the same shape as the chemical messengers that are being sent, so they bind to the active sites on your nerve cells and stop them from bouncing back again to make your signals send properly. So that's an example of where we use the chemistry of a substrate